So with that, we're gonna move on to our next phase of this tech session, which is the bioinformatics, session. Chris Taylor is gonna introduce the session. He is the core director of the BBC core, and I'm eager to hear what he has to say about the bioinformatics core.
All right, so it's working. So I'm the director of the Bioinformatics, Biostatistics, and Computational Biology core, and we're going to have a few people present just like the MCBR core did. And I'm actually based in New Orleans at LSU Health Sciences Center in the microbiology department. So I'm going to give a little introduction and overview of the BBC core, and then we're going to talk about our partnership with Pine Biotech and the Tauber Institute for some educational programs and analysis pipeline support that we have. And Emmanuelle is going to talk about RNA-Seq analysis and Ingenuity pathway analysis. And then finally, we'll discuss some of the computing infrastructure available and computing boot camps that are available through our high performance computing group. And then lastly, there'll be box lunches and Q&A with the two cores. So the mission of the BBC core is fourfold. We want to assist with connecting appropriate collaborators and mentors. So our LBRN researchers who need collaborators with specific expertise in data analysis or need mentors for their projects. We provide essential computational analytic expertise to LBRN researchers, and we aim to educate the researchers in computational techniques, and this is done through the partnership with Pine Biotech, the educational programs that'll be discussed, some computing boot camps that are done through the HPC group, and some other initiatives that we have. And then lastly, we aim to expose the LBRN researchers to the latest analysis research methods, and that is primarily through our annual conference on bioinformatics and computational biology, which I'll discuss. So as you know, the LBRN, our institutions are distributed throughout Louisiana. In terms of computational analysis, I feel that it's best to pair people up, not necessarily based on their geographic proximity, but based on expertise, as a lot of the data analysis can be done and meetings can be held remotely. The core organization, so the leadership team consists of myself, the core director, Dr. Erska Savek, who is at Shreveport. She's an associate director, Nayong Kim, who is also associate director, and our new addition, Dr. Emmanuelle Ruiz, who is an assistant director and is assisting with the Pine Biotech pipelines and also IPA analysis. John Kibdo is our computer manager. And then we interact primarily with the LBRN institutions through the core liaison officers, which are listed below at our 8 institutions. So the idea is that each liaison officer at each institution interacts with their researchers at their institution and kind of communicates up to the leadership team what the needs are, and we work through them to provide support for the researchers. So the core liaison officers, they're appointed for 1-year renewable terms. They provide, as I said, a point of communication for each campus. They identify the needs of the campus in terms of analytics and their LBRN faculty, and they communicate those needs to the core leadership team. We're holding monthly core leadership meetings with all of the core liaisons. And we also have a core bucks program, which is also available for the MCBR core as well. And that provides very small amounts of funding for things where you just need a little bit of preliminary data. And so that, again, the process is the researcher at the LBRN institution will get in touch with the core liaison for the appropriate core. and initiate a core bucks application, which then gets reviewed up through the chain. And those, the core bucks program, it provides a pool of funding for our members and their investigators to apply and receive our core services to assist with data analysis. So all reasonable requests that we get are typically funded, and you can utilize those funds for services that are available with any core that has a schedule of listed fees. So examples of those, the Gene Lab here in Baton Rouge. I have a microbial genomics resource groups, which has our own schedule of services available at LSU Health Sciences in New Orleans. Erska is involved with the bioinformatics core in Shreveport that she runs. And then we also have the Pine Biotech analysis services through our LSU partnership. So here you can find this on the internet, but we have the Core Bucks flyer available. And again, it just kind of goes through some of the process of applying for the core bucks and some of the things that are available. And for more information, there's a course link on the LBRN website. And again, you, as a member institution, you connect through your core liaison officer, of which are listed here for each of the institutions. These are for the bioinformatics computational biology core. There are also appropriate core liaisons for the MCBR core as well. And then if you have technical questions regarding the core services, our assistant director, Dr. Emmanuelle Ruiz, is the primary point of contact for that. And you know, if you have computational questions, you can get in touch with Dr. Nayong Kim as well. So if you have administrative questions regarding core services, Brent or I are your points of contact for that. So we run a annual conference on computational biology. We held the 9th annual conference on computational biology last April. It was a return. We basically had to cancel our 2020 conference, and then we did virtual conferences from 2021 and 2022. So we held this meeting virtually. We were able to get some very high caliber speakers since it was virtual, but we are planning for the 10th annual conference, which will be a return to in-person and is planned to be held joined to the 2023 NBRE regional meeting, which is tentatively going to be in mid-September, 13th through 15th, with the regional meeting to follow on the 15th to 17th. And that'll be at the University of South Carolina. So I've provided some lectures on informatics that are available. The links are here. They're available on the cores. Slide sets, videos, and other lecture material are also available for download for those lectures. Those lectures were used when they were given and joined with some courses at the member institutions, and they persist available for utilization of those institutions in the future. I'm going to skip that. Emmanuelle is going to talk about the Pine Biotech program. It's an educational initiative that we partnered with Pine Biotech and the Tauber Bioinformatics Institute in 2018. It's been expanded since then, and the students who participate in that program are encouraged to present their project posters at our annual meeting, and we've expanded that into a full partnership. So I'm going to hand it over now to Emmanuelle. She's going to talk about the sequencing and Ingenuity pathway analysis.
Good morning, everyone. So I'm going to present you a bit what the Gene Lab Biomed provide as services for sequencing, and a little bit of description of the Ingenuity pathway analysis IPA. So before to go further, I want to present myself, as we will have the occasion to work together if you have a bioinformatic needs. So as you could understand with my accent, I am French. I have had a background, a PhD on molecular and cellular biology and a pharmacy. And I have, so during my career, self-learned bioinformatic tools. As a lot of people, I think that on biology, when we have the needs of bioinformatics, we are here with our questions that are very undefined because we just don't know exactly what we ask, and we don't know the tools that we need for responding on question. And my relationship with bioinformatician was quite good, but I didn't succeed to explain to them what I wanted. So I decided to learn by myself how to do what I wanted to do. And I fell in love with bioinformatics and decided to continue to work on it. And so the field of research that I have the occasion working was cancer, particularly cancer, breast cancer, thyroid, and melanoma, has a basic but also a translational axis with discovery of new prognosis markers, and as well as genes or microRNA. And I also work on IBD, in inflammatory bowel disease, currently, so since June now, I have joined the team of Dr. Kusulas in the pathobiological science department of the school vet. I am, as Dr. Taylor has said, as associate director of the LBRN BBC core, where I will provide bioinformatic help and collaboration on sequencing, gene expression analysis, but also other needs. I am also the coordinator of the Biomed Gene Lab and Omics Bioinformatics training program that we will have the occasion to talk about in the next slides too. So the Biomed Gene Lab provide some next generation sequencing services, particularly RNA sequencing, but also DNA, sheep and single cell sequencing. So with these services, we perform data processing thanks to the LONI HPC system here in LSU, but also the Biotech bioinformatic platforms. And from these files obtained from the sequencing, with different packages, we obtain the normal gene expression files. We have the possibility we have access to the classical software algorithm used for the processing in a way that are standardized, and also that we can customize them to respond a maximum of your on your question and conditions. And after, finally, the fun begins with analysis by itself. We begin by the differential expression gene analysis. We will obtain the genes that are differentially expressed between your condition B and condition A. And also, we perform enrichment per square analysis. I will describe more with the IPA in the next slide. But we can go further than that. We can look at macroenvironment analysis with different algorithms that give you the enrichment of immune cells or other macroenvironment cells that are around your condition. Also, we have the possibility now to validate in public and online database as the sequencing RED archive or the GEO repositories that give us a plethora of data sets that that can be used to validate your own work. There is a different platforms that gathered public samples, has a Cancer Genome Atlas, the GTEx, that's the genome tissue expression something, and the Cancer Cell Line Encyclopedia that are popular, but there is also other ones. And the next step is also to have a translational perspective of your project, is how much what you have found on this analysis have a clinical on outcome impact on human samples or human samples, but yeah. And so we can perform clinical and outcome parameter association, and also go further to see how much what you have found in this basic axis can be used in the translational and clinical axis with discovery and validation of diagnosis, prognosis, or therapeutic markers. So that is the fun. And so I'm gonna go, we can do also a lot of different things, and globally only our imagination can block us to go further. So I'm going to describe a little bit more about the enrichment pathway analysis. So we have access online on different databases that are free, like the KEGG, the gene ontology, the reactome. They can be used with the software R or Python, R, that is sure. And also there is platform in the web that use them and are very easy to use. But there is also the Ingenuity pathway analysis, the IPA. The IPA is a web-based bioinformatic application that allows researchers to upload data analysis results from high throughput experiments such as microarray or next generation sequencing for functional analysis, integration, and further understanding. This includes both microarray, but also RNA gene expression, but also microRNA, SNP, metabolomic and proteomic data. So it's not only your gene, but it's also a global analysis that can be used as an input. In general, list of genes or chemicals also can be analyzed using IPA. It has also a search capability for information on genes, protein, chemical, and drug, and allow interactive building of networks to represent biological systems. So it's a quite wide, deep database that is highly cited, there is more than 4,000 citations on PubMed. And also when we wish to publish in high impact factor journal, is highly recommended because it's very powerful database and very straightforward and also robust. So I'm going to describe the basic use of IPA with enrichment pathway analysis. So the input is your gene, your differential expression genes or proteomic or metabolomic or microRNA. And you have your fold change and your p-value, and you insert that on the software, and they're going to present different analyses. The first one is a pathway analysis. They give you a list of pathways that are enriched with p-values and adjusted p-values. You can customize the plots more or less as you want, but they have a lot of possibilities. What is interesting on IPAs that do not do most of the free algorithm is that it gives you an estimation of the z-score that give you an estimation of how much this pathway is activated or is inhibited. So you have he give you the list of the enriched pathway based on the differential expression genes, but also it gives you an understanding, an estimation of how this pathway behave in your condition, how much is activated or is it inhibited. And that is quite particular of IPA. After draw this beautiful plot, it depends on how you like it, but I find that nice. These plots also are quite interesting and quite unique of IPA. Whereas first, they show you a very wide and deep database of relationship between proteins, from regulation to interaction to modification to translocation, etc., etc. And it gives you, so it colors the protein based on the corresponding genes, if it's upregulated or downregulated. And finally, it gives you also how much the genes is the protein can be activated. The edges, how the edges between two proteins, how the behavior is it? Is it an activation or an inhibition based on the color of the edges? That is quite interesting. It is not found in the KEGG analysis or gene ontology. It's very much unique of IPA, and it gives you a better understanding about how this pathway and how subpart of this pathway is are behaving. So, for example, on this network of autophagy, we have a lot of mechanism here that lead to an activation of all these proteins. And we see that this little part actually has, this one is downregulated. What is below him or downstream heme are inhibited. And also it show you in the yellow part what actually is not consistent with literature. So I say he, but it's not able to estimate if there is activation of inhibition. It presents you other plots, has a bubble chart. We give you that present the pathway based on their categories, cell cycle, immune system, etc., etc. Give you the overlapping pathway. So, for example, on this case, we see there is a bunch that are strongly together. They have a lot of genes in common, and they have these three, the ones that are a little bit alone. So it gives you more information and an overview of your analysis. And finally, a summary graph. The second aspect of IPA that is quite powerful is how it builds network of the protein or genes with interaction and regulation. Also, it gives you the, I will say, it score everything with p-value. So it gives you a list of network from the strongest network to the lowest. And you see that some are overlapping, and they show you how they overlap. You can present a network in a different setting, had a hierarchical, a radial, and also a subcellular that give you an idea about what is happening according to the cellular localization. The third aspect of IPA that is very interesting is estimation of upstream regulators. You have your genes, proteins that are potentially deregulated, and he's gonna estimate the upstream regulator has transcription factor, enzyme, metabolite, hormones, but also therapeutic drugs. He have a wide database of this non-regulation, and based on the gene expression, he's gonna estimate the activation and inhibition of the upstream regulator. So for example, if I have a gene A that is downregulated and is known to be activated by the transcription factor A, this transcription factor A is gonna be also inhibited. And so based on this logic, he's gonna estimate the activation or inhibition of the upstream regulators and give you networks of regulation where we have, for example, in this case, the doxorubicin, that is known to activate or deregulate this set of transcription factor or other regulators that secondly regulate or control the subset of your list of DEG. So it gives you a wide information only in one software. So it's very much interesting and powerful. Finally, we'll go very fast. We have also access to the disease and biological function that give you a little bit an overview of your analysis above your own condition, and also give you an access to what is happening in other analyses from the IPA database, where they correlate your analysis with others, and you can estimate more or less if you have your analysis can be validated in other IPA analysis have been performed in the past. And you can also, with this activation plot of a single pathway, where you see me personally, I don't see it, but there is a green dot somewhere there, where that is your own activity of your pathway, that you can see it among the other IPA analyses that have been performed. And so I will be glad to help you with your IPA analysis and help you the best as I can. And but we're gonna talk now a bit further on the next presentation about the next winter program training that we partner with Omic Logic. And thank you for your attention.
Fortunately, Elia couldn't make it today, so Emmanuelle's going to be presenting for Fine Biotech. But she's well versed in the platform, and looking forward to hearing what she has to say.
I would like to present you what Fine Biotech and Omic Logic propose for the LBRN networks. So Omic Logic is a platform of training and teaching on bioinformatics that is available from anyone. And when I say anyone, I mean anyone that don't know anything about bioinformatics. They propose a variety of very well done.
Oh, sorry. Trying to get this to go away.
Thank you. Of bioinformatic application from genomic, transcriptomic, metagenomic, epigenomic, but also data science of how to code on statistical analysis, machine learning that can be used for biomedical data. And they propose practical and effective use with R and Python that are two principal software used in bioinformatics. They organize their training on 3 different steps with interactive sessions, but also self-guide studies materials that you do by yourself and practical assignment with project where we can use online dataset and work with them and learn how to use all these beautiful tools. So this winter or spring, there will be a virtual training that have 5 different courses that are from the introduction of multi-omics beginner with after an intermediate the use of R on we'll go back on biomedical data or on R, but also on Python, and after on more advanced chemoinformatics for biomedical drug discovery and machine learning for specific health informatics. So that are the different courses that will be available for this training. And a live orientation will be held the 30th of January at 3 p.m. And if you want more information and to register, please go to this edu.omiclogic.com LSU-biomed. And you will have access to the registration and also more information. And so that's it. So this program is sponsored by LSU Biomed and is available also not only for the LBRN, but for the NAPI and other people that want to learn more bioinformatics. Right now, and I'm saying that because we have several questions about that, there is not university certification or to have credit, but it's a great tool to have a better understanding and practice. So we don't care. And we don't care. So to get it started, you will have access to introduction of bioinformatics, different application from not only on human biology, but also different other aspects. What you have access also only when you register, so don't need to further access to different courses and teaching about bite and molecules that give you an understanding of DNA, proteins, and other tools that they will use for the training, and a little bit of R and Python use. And after that, they will describe globally the genomic, transcriptomic, metagenomic, single-sex sequencing, epigenomic, and also some case studies to show how much these tools can be used on the day-to-day research life. You will have courses. Every time you do a course, you have points. And when you will have 10,000 points, you have access after to project that will be specific project, specific project that you show, that will where you will have specific data set, a list of things to do on R or Python to work on this project, and have after obtain result and visualization. For that, you will use this the Tober T-Bio platform that gathered all of algorithm in order to analyze the data, and they will give you access to it to perform that. This platform is okay, maybe something here. Have a wide range of utilization from transcriptomic, DNA, RNA, mass spectrometry, structural biology, data mining also. So what is interesting in the data mining, you already have your analysis, but you just don't know exactly how to visualize them or to analyze them. So you can do here supervised, unsupervised, we do that again. Unsupervised and supervised analysis, PCA, etc., etc. So that is quite powerful. And you have also on single cell analysis that is quite new now, and you can use it as to process your sequencing, but also to analyze further, and etc. So it is quite powerful tools. It's standardized, so you don't have to play with a lot of parameters, and it's reproducible. So after the second training that will be proposed is an introduction to data science in R, where you will learn how to use R in terms of how to load and the data, how to process and visualize the data, and different also other tools that are used on R to analyze the data, has PCA, differential expression gene, or clustering that are quite, R is quite powerful for that, and it's quite fun actually to learn it. You will also, so that we present here for the R software, but it will be more or less the same thing as say for Python. That is another free software. And on all the cases that they will use as example, it will go from precision medicine on all these different possibilities, biological production, for example, epidemic and antiviral. You have the Ebola, but you also have the COVID-19 examples to work on. So it's not only, it gives you also a possibility to go out of your own research field, and is interesting. And so I want to remind you, so the live orientation will be January 30th at 3 p.m. They will give you a lot of information of how to register, but also to log on the sign up on their eduemiclogic.com, and all the information that need to be done. So the LBRN is offering this training, so it's free for you. And you can take for this semester only one of these 5, in order that you have the time to focus on it and complete it. And that's it. Thank you.
Okay, and we're moving on to Nayong, Kim, and Elia, who will be talking about some of the bioinformatics infrastructure we have here at LSU as far as high performance computing and the different software that's provided. Thank you.
Hi, my name is Nayong Kim. I'm admin core of the LBRN, also the BBC core of the LBRN also, and then also I'm an assistant director of the Center for Computational Technology, LSU here. So we are more focusing on the science, supporting science through the HPC. So I will steal the 1 minute from the Lian's presentation actually, and Dr. Lian also assistant director of the Loni HPC. He will discuss about more in detail. So today I will explain a little bit clearly explain about how we support 5 categories from the BBC core. The first one is Illumina base-based sequencing hub, which is a license, we have a license in the GenLab here, best core. So there's one of the way we are supporting, and the other one is CCT, LSU, LONI, HPCs that will be presented by Dr. Liang, and PineBio, which is just mentioned about the Dr. Ruiz, and the IPA also, she just mentioned about that. And the last one is a network attached storage system, which is we are store the data to support people. So we have two different kind of HPCs. One is a LONI, which is supporting statewide campus. So all people here, researchers, students actually can use LONI HPCs. The other one is the LSU HPC, which is supporting this campus, LSU Baton Rouge. But if we have a collaborator here, then any people can use that also. So we have several options to use it. One of the interesting the computing system is a deep bio, which is LSU HPC. The Liang was is the PR of that the computing system, which is granted by NSF. This is focusing on the deep learning machine. So also we are providing the LONI HPC allocation, which is a computing hours. So if anyone who's here interested about using the LONI HPCs, please contact me. Then we already, based on the collaboration, we already have a million computing SUs, so we can provide it to you without any the cumbersome stages. The other one is a bootcamp. We are providing the bootcamp every year. This last year was the 5th bootcamp. It was virtual bootcamp because of COVID-19, but we are providing the bootcamp maybe this year is in person. We are not decided yet detail, but we will have that another bootcamp. This will come, covering the bioinformatics, also the deep learning.
For the bootcamp, there are recordings of the bootcamp, right?
Yes.
That people can go back to.
Yes. Yeah, but bootcamp is they're more enhanced, the training. Yeah. So next one is the network attached storage, which is NAS. We have several NASs here, which is managed by the John Q, famous person here. So if you have any question, so please contact him. But we have a kind of internal, the policy, which is we cannot store the data, your data entire life. So basically, we're gonna stay the data up to 6 months initially. Based on the request, we can extend that, but that's kind of internal, the policy. And we are focusing on live data, which is using data, not the passing data. Okay, that's all my part, and then I'm gonna pass it to Dr. Liang.
Can everybody hear me okay? Okay, cool. Thank you, Nayong. My name is Lian, I'm Assistant Director for LONI HPC. So as Nayong mentioned, when we talk about HPC, there's actually two parts, one for LSU and one for state, which is under LONI, because this is, I guess, a statewide event. So I'm going to cover the statewide infrastructure today. So as you might or might not know, LONI, LONI, stands for Louisiana Optical Network Infrastructure. Used to be initiative, but after being initiative for like a decade, people think, well, probably it's time to change the name to infrastructure. So since 2006, actually for the last 16 years, LONI has been operating the research network backbone for the state. So connect all the research universities and actually a few other institutions, organizations as well. For example, PBS, the LPB uses LONI network to move their media data across the state. And also LONI is your conduit to other national research networks. So on top of the network, LONI also provides HPC high-performance computing resources to researchers at LONI member institutions and their collaborators free of charge. So that's a service that I'm responsible for. Yes. Okay. So we have two clusters right now, QB2 and QB3. Combined about 600 servers, more than 17,000 CPU cores, about 65 gigabyte of memory, 1.5 petabyte of storage. So we are actually, the QB2 cluster is kind of old. I think it's 8-9 years already. So we are actually in the process of acquiring another cluster, QB4 from the name. You can imagine how creative we are. But nonetheless, it will have almost 600 nodes, and it will triple the number of CPU cores, and it will have about 165 terabyte of memory and about 7 petabyte of storage. And I didn't put it here, but it will also have about 150 NVIDIA A100 GPUs, which is technically not the latest generation because Hopper came out, but right now it's still, I guess, what you can buy is kind of still the latest GPU. Will be available. I think this machine should be up and running by the end of the year, finger crossed. So on top of the access to the compute CPU cycles and storage, we also provide services like we can troubleshoot issues for you on the clusters, and we can provide some consulting services for your applications as our expertise allows. And then we provide training, so we do the bootcamp, like Nayong mentioned, also do weekly trainings on various topics related to HPC, and also we do grant proposal support. So basically, if you need a letter of collaboration, you need a letter of say commitment, so you have the resource available, or even you need some language for say the facility is available to you, a description of what Lonnie HPC resources look like, and we do those kind of things as well. So if you have that kind of need, just shoot us an email. So to use the Lonnie HPC clusters, it used to be the case that you need to be familiar with command line interface. So basically that's where you need to type in all your commands, right? So we understand that it is not for everybody. It's challenging for especially the younger generations, and also just to acquire that skill set, the learning curve could be quite steep sometimes. So we actually recently implemented a web-based HPC portal. So it's browser-based. So if you go to the URL on the bottom of the screen and type in your Lonnie user credentials, you'll be able to log in this portal called Open On Demand, which is developed by the Ohio Supercomputing Center with NSF funding. So and there you can do a lot of things. You can browse the file, manage your files, and you can run jobs. And also you can you can run interactive applications such as Scrivener Notebook and R Studio. And also we are so basically using this platform, it has some capability allow further development. So basically we are also developing new applications. I put 2 examples here on the left. AlphaFold, which you might not know, is a deep learning based software that does pertain 3D structure prediction with very high accuracy. So the other one on the right, cell render, I think this is a single cell RNA sequencing pipeline. So these are you see the interface looks pretty similar. So basically we kind of use the same template and basically try to kind of turn the applications into pipelines into a web form where you just put in all the parameters instead of typing all the commands. You can just put in on the same web form and hit the submit button, and it will run on the HPC platform. So some numbers for the year 2021. I haven't got the numbers for 2022 yet. But in 2021, we delivered 80 million CPU core hours to users. I think the number will be close to 100 for the year of 2022. We have more than 100 active PIs, research groups on the Lonnie clusters from 12 institutions across the state. Actually, the number, if you count the collaborators, the number will be higher because, like I mentioned, so the collaborators are eligible as well. So, but when we do this, we kind of lump the collaborators, into the institutions they're collaborating with. So we did 26 weekly training sessions. This is on top of the summer bootcamp. So, and also across the state in the year of 2021, there are 10 courses taught, I think, in 4 or 5 different institutions using Lani HPC resources. So there are a lot of different research projects on Lani, and it's could as big as a study of the universe, could be as small as a study of molecules or atoms. So the next few minutes to wrap us up, we'll just go through very quickly some of the projects that are using Lani HPC. I'm not a domain expert, so I just did my best to glean from this description and the report the researchers submit to us and try to categorize them. So if you see anything stupid, doesn't make sense, silly on the next few slides, the error are totally mine. And I'm going to just talk about the biological, biomedical, bioinformatic, kind of research. So there are a few groups are actually doing AI deep learning related research on and by the way, it's not a comprehensive overview. So I just choose a few that I'm more familiar with. So there are a few groups on the Lani clusters are doing AI deep learning related research. For example, Dr. Carmichael from Pennington, they're trying to see how they can use deep learning model to create other type of scans from one. So basically, if you have your PET scan or MRI scan, they're seeing if they can use AI to create, to synthesize other type of scans from the scan you actually did, right? And there are other 2 groups from Tulane using PyTorch to develop neural network models for various purpose. And I think there are people also using AlphaFold for protein structure prediction. And also biostatistics. I don't think I will even try to understand what they do. So we have groups from HSC at New Orleans and also Pennington, or I think all of them use R. Some of them use, I think at least one of them use the portal I just mentioned, RStudio, to run their jobs. And some single cell sequencing, Dr. Wang from HSC on New Orleans and Dr. Coase. I think Dr. Wang, both of those groups, I think started quite recently, I think last year, a few months ago, doing some single cell sequencing analysis and other type of genomic data analysis. Dr. Bose from LUMCON, I think he's a adjunct at Tulane as well. So basically, he's doing some metagenomic study for marine biology. And Dr. Franz from ULL, I think doing similar genomic data analysis for some marine lives. And then Dr. Rowe is from the Tulane National Pyramid Research Center, and she's doing genomic data analysis where data comes from their nanopore sequencing. And we have quite a few folks who are doing phylogenetics. This is not a comprehensive. I think I skipped a few folks. 2 from Dr. Pillar and Dr. Wright from Southeastern, and Dr. Sackett and Dr. Poolers from ULL. They're doing various different kind of phylogenetics studies using Lani HPC. And then I think that's the last slide. So these are not so those are what I just showed. Mostly they're just data analysis, right? These two groups from Tulane, they're doing kind of simulation. This is more like, a traditional HPC simulation. Basically, for example, the one on the right, Dr. Fujioka, he's basically doing fluid dynamics, just trying to see how the mucus, how the fluid in your lawn would flow under various conditions. And I think they write their own code with C++, Python, and MATLAB. So this is the last of my slide. What you're looking at here, actually, this is the data center at downtown Baton Rouge. This is the information services building of the state. And they're looking at the black thing, there, the QB2 cluster. So that's what the supercomputer with the cluster looks like. So like I said, a lot of research projects going on. So I put a question mark there. So hopefully when I do something like this the next time, your research project will be at the place of the question mark. So I think we have a few minutes left. Yeah, I mean, I'll be glad to take any questions. I know this is a very quick overview. Yeah.
The machine learning stuff that you showed. I have a two-part question, I guess. What's the types of frameworks that are supported? Like you have Keras and PyTorch and things like that.
Yeah.
The second part of the question is you also have GPU processing.
Repeat it so that.
Okay, all right. So the question is first, what kind of deep learning AI deep learning software frameworks available? And second question is, do we have GPUs? And the answer to both of them are yes. Well, first question should be yes. First question is so PyTorch, Keras, TensorFlow, those we have them. Some of them installed centrally so that, after every got account and log in, you will see them. But because AI deep learning is a very rapidly advancing field, I mean, they constantly churning out new versions. So what we mostly do is we will support installation in your own directory with your chosen version.
Okay.
We have a documentation and also we can provide help. And then GPU, yes. So right now we have the B100 GPUs. And like I mentioned, the next one. So we are in the process of acquiring it. I think the quote has been created, but hasn't been approved by purchasing yet. But and there will be time before, when the purchase order PO is cut and when you can get them.
Next fiscal year.
Definitely next fiscal year, hopefully not next the calendar year. So that's what we're hoping for. But yes, so on that machine, we will have about more than 140 A100 GPUs.
Yeah.
So that hopefully will
Thank you.
yeah. Yes.
You went quickly through the open on-demand interface for AlphaFold. Is that available now or soon?
Soon. It's
Repeat it.
it's available on in the alpha or beta versions on the LSU side. That's where we did the development. So we will move it to the line side.
Actually, it's possible to use now. If we, as the concept was, we can, put you as a collaborator, then you can use it.
And also, so let me say this. We do have, so I think Brent here is, so I can run some, right now, if you are okay with the command line interface, you can use it. So Dr. John from SHU, actually, he is using it. It's just, if you want to wait for the open up and the web portal to be fully functional, then it will take a bit of time.
You'll probably finish it before I learn.
Probably. All right, thank you for your attention, and I'll be here for lunch. So if you have any questions, want to chat about, what you want to do but not sure about, if you say, by all means, seek me at lunch. Thank you.